O tēnā koutou katoa, ki te whare e tū nei, te papa o tā koto nei, nā rangatira hoki, tēnā koutou. He mihi kauana ki te mana whenua o tēnei rohi, ki a koutou me a koutou uri, e muri aki nei, tēnā koutou. Tēnei au te paukoko Māori o tākou whakaihu waka, he mihi atu nei, ki a koutou, tēnā koutou, tēnā koutou, tēnā koutou katoa. I am Jacinta Ruru, Deputy Vice-Chancellor Māori here at the University of Otago. And on behalf of the senior leadership team, I welcome you all here this evening for this very special event to mark and celebrate the promotion of Professor Janice Lord to our highest academic position. He mihi nunui kia koe, Janice. I acknowledge all our colleagues and visitors here in person and those online who have gathered together to celebrate all you have achieved in your remarkable career to this point. This is a special occasion for you, Janice, and for us all. And I especially acknowledge your partner, Ashley, and your two sons. I believe in the audience here this evening is your son, Oscar, and his partner, Eilish. Both have studied or studying here at the university. And their son, Archie, who looks like he's right here in the front row, and who's your first grandchild and is nearly 2. And joining via live stream from Wellington is your other son, Tor, who is currently studying architecture at Victoria University, and I very much hope that he has been able to join us this evening online. This occasion this evening represents years and years of high-profile research, teaching and supervision. And as an expert in botany, Professor Lord has contributed enormously to new knowledge in this country and internationally. And one of the roles of a university is to help positively transform society, and your disciplinary work in the science of botany was truly remarkable and has deservedly led to many recognitions. Professor Lord, I'm inspired by your research, teaching and service, and as one of your much admired colleagues, it is my greatest delight to stand here with you this evening to mark this substantial occasion in your stellar career. I'm now going to hand over to Pro Vice-Chancellor of Sciences, Professor Richard Barker, to more fully introduce you. So our warm welcome to you all this evening. Tēnā koutou, tēnā koutou, tēnā koutou.
Tēnā koe, Professor Ruru. Tēnā koe, Professor Lord. And Professor Sika Pautonu and Professor Blakey, tēnā korua, Professor Olovich, tēnā koe. Friends and colleagues, tēnā koutou, tēnā koutou, tēnā koutou katoa. It is my pleasure to introduce our speaker tonight, Professor Janice Lord from the Department of Botany, to give her inaugural professorial lecture at the University of Otago. As indicated by Professor Ruru, Janice was promoted to Professor in February this year, and tonight we get to celebrate this significant achievement. Born and raised in Christchurch, Janice went to Banks Avenue Primary School and then Avonside Girls High, and highlighting the importance of female role models, she was most inspired by her wonderful science teacher, Mrs. Ridd, and maths teacher, Miss Crest, during those formative years. Janice's employment path to the university is an interesting one. She started out as a nurse at Southern Cross Hospital in Christchurch before moving into clerical roles, first at the Municipal Electricity Department and then State Insurance. Janice then joined Christchurch City Council as a park caretaker and then moved into a research assistant role at DSIR Grasslands at the same time as studying for her BSc Honours in Botany at the University of Canterbury, where she was awarded the Bill Makepeace Prize in Botany. This was followed by her PhD in botany, also from the University of Canterbury, and while doing this, she spent some time with the Department of Conservation, carrying out an environmental impact assessment of the Speight's Coast to Coast endurance event. Completing her PhD in 1992, Janice undertook a postdoctoral fellowship at Macquarie University before then becoming a New Zealand Foundation for Research, Science and Technology postdoctoral fellow. And in 1996, Janice completed her roundabout journey to join us here at Otago as a lecturer in our Department of Botany. And so tonight we celebrate her rise through the academic rank from lecturer to professor. Promotion to the position of professor at the University of Otago is far from easy and requires excellence across all three academic domains of research, teaching and service. Professor Lord's excellence in research has been recognised by the Royal Society of New Zealand Te Aparangi, first with her selection for the Leonard Cochrane Memorial Lectureship in 2015, and then when she was became a member of the list of 150 impactful women in the history of New Zealand science that was published in 2017. Impact is a standard that we all aspire to in our research, and how better to make an impact on one of the leading causes of the day than to be the leader of the 1 Billion Trees program funded by the Ministry for Primary Industries, which aims to make a meaningful difference by removing atmospheric carbon that is a peril to our lives and communities today. Janice has not only been an impactful researcher, but she's also been a productive one, with more than 120 peer-reviewed articles and 7 book chapters having been published. And these represent projects that incorporate both botanical science and Mātauranga Māori, and many of her publications are co-authored with her postgraduate students. Janice has supervised 34 Masters and 16 PhD students to completion, and for this has been recognised through a nomination for the University of Otago Supervisor of the Year Award in 2017. A hallmark of Janice's long teaching career is that she connects the academic study of botany to the real world of our students, and this is demonstrated in her focus on topics such as sustainability, restoration, and ethnobotany, all of which are informed by her research. Janice was involved in the establishment of the ecology program in 1997, the genetics program in 1998, helped redesign the botany curriculum in 2013 and 2014, and most recently helped us develop our agricultural innovation program. She has developed, convened and or taught undergraduates at all levels in botany, ecology, genetics and agricultural innovation, and has also taught in other divisions in the Division of Humanities and Health Sciences. She has obtained a number of grants for teaching innovation and has championed Māori content in courses, including conducting a review in 2018 of the use of Te Reo Māori, Māori cultural concept and Mātauranga Māori in the undergraduate botany teaching program. And this brings me to the final domain, academic domain of service. And it is notable that Janice's research leadership has gone hand in hand with her service to advising government and non-governmental organisations. For example, the Ministry for Primary Industry, the Ministry for Environment, the Parliamentary Commissioner for the Environment, and the Queen Elizabeth II National Trust. Janice's high-profile connections with these organisations benefit the university and lead to important engagement, such as the recent visit by the former Minister for Climate Change, James Shaw, who visited the Department of Botany in 2023. Internal to the university, Janice was the Ecology Program Deputy Director from 2010 to 2014 and worked with our Office of Sustainability to develop Te Kauka Sustainability Framework and on our air travel project. As with her research and teaching, Janice has been recognised for the impact that she has on our local and professional communities. She was one of 125 women featured in a Ministry for Women-funded Suffrage 125 exhibition at Tuhurua, Otago Museum in 2018, and in 2014 was awarded a Fulbright New Zealand Award for Excellence in Educational Achievement. I think that's enough from me. It's now time to hear from Professor Lord herself, one of our stellar women in sciences here at the University of Otago. From flowers to trees, botany in a changing world, please join me in welcoming Professor Janice Lord.
Tuatahi ake name te mihi ki Professor Ruru tēnā koe. Me te mihi kia Professor Barker, tēnā koe. Ko te mea tuarua e rere a te ana te mihi kia koutou katoa. Kua hui hui me nei e tō koutou whahi wai e tēnei rā. I'm shaking. What I'm trying to say is thank you to these wonderful people for all the fantastic stuff you said, and I'm so glad people turned up because the weather's not very nice out there and I was getting a bit worried that nobody would turn up. But here you all are, and it is so reassuring to see so many smiling faces, and I feel so much better. So, okay. I know so many of you, but ko toku pepiha tēnei. He tangata tiriti ahau, ko Kautarani ta whakapaparonga mai, ko Thomas Lord tōku mātua no Northumberland ia, ko Marjorie Makoni tōku whaia, ko Oti Poti tōna kainga. Ko o te tahi te whenua tupu I tipu aki au I te rohi o kaitahu, e raro I te mare o Aoraki. Ko Janice Lord toku ingoa, no reira tēnā koutou, tēnā koutou katoa. And in my pepiha I'd like to acknowledge the origins of my family, but also that, you know, I grew up in this in Taiwaiponamu, that's kaitahu rahi, under the shadow of Aoraki. I will start with just acknowledging my family because it's kind of accidental that I ended up in university and I ended up here, as Richard pointed out. I had a bit of a circuitous route. I've been to these IPLs before and it amazes me that some people just naturally move into tertiary education. I certainly didn't. My childhood was very plant-based though, and I'm very grateful for that. So my father was ahead of his time. He was an avid composter. He would go out and grab other people's waste and compost it. He insisted on wearing second-hand clothes. My mother was embarrassed of him, but he grew the most wonderful vegetables and fruit, so we were very self-sufficient. We didn't have a lot of other material possessions, but we had best vegetables and fruit ever. And we went on these incredible camping holidays, as you can see from the old Hillman Comic Cob and the canvas tent, which shows you how old I really am, and the terrible faded photographs, which gave me an opportunity to just be feral in nature, and I really thank my parents for that kind of upbringing. And often we went on holiday with my grandparents, that's the top picture there, so my mother's parents, and that was another level of plant-based knowledge because my grandfather as a young man worked in flax mills down in south of Otago, and he and when I was thinking back about it, and he really was conveying to us the tikanga around harakeke because he had what was in effect a small pa harakeke up the back of their property in Hampden, and us grandkids were not allowed to play in there. We had to respect the plants, and he would use them to make twine. So he had picked up those skills from his time spent harvesting harakeke. So I was a feral child. I would ride my Rally 20 with my terrible haircut. I thought this would make the students laugh. I would ride my Rally 20 up around the Port Hills in Christchurch, exploring. But our family didn't go to university. We just weren't that sort of people. I had some random older cousins that had trained as nurses, and there was one much older cousin who had a PhD in geology, and he might as well have been a funny green man from Mars for all I could relate to that. So I left school at 16 and ended up working in an office. It was only when someone in that municipal electricity department suggested that they pay my way through university to get an accounting degree that actually made me think, oh, you know, someone like me could go and get more education. That was an interesting idea. And it took a bit, but I did end up at the University of Canterbury. But my upbringing had very much trained me to be of practical use. And my mother used to say, all those boffins in their ivory towers in the university, they're no practical use whatsoever. So I enrolled in a Bachelor of Forestry Science initially, thinking, well, that's practical, growing trees. My life changed because there was a young woman, and whose name I never learnt. She must have been a postdoc. She was wearing a white lab coat. She gave a lecture in first year, and she lectured on alpine tree lines, and I've discussed this, you guys know this story. I was captivated by what she was saying, but I think quite rightly, last time I discussed it with my sons, it was also seeing someone I could relate to was life-changing. So the first lesson of the day is that you do not know the impact that you have just by being yourself and representing. That's the first lesson of the day. So my PhD, I spent 3 years and a bit wandering around Arthur's Pass National Park. That's 80s perm. We're going from 70s shaggy to 80s perm. I fell in love with seeds and plants and reproductive ecology, and then that led quite easily to a postdoc in the lab of Mark Westerby at Macquarie University. So I'm going to start talking about research. I'm going to start with seeds because so many things start with seeds, and we use seeds to symbolise the start of things as well. I particularly love Ritidosperma. It's one of my favourite genera of grasses because they're so pretty. Aren't they pretty? Yeah, they're very pretty. But seeds encapsulate that alienness about plants. So a seed actually contains the next generation, a genetically distinct individual embryo that's packaged with parental resources. So there are resource allocation decisions about how those resources are, how much resources go into that seed. And then the outside parts of a seed are actually parental tissue. So the parent is not only protecting the seed, but potentially controlling how and when it germinates, as well as controlling its size and affecting how it can disperse as well. So they're really interesting sort of packages that combine all of these trade-offs and resource allocation strategies that go and a really important part of plant ecology and reproductive ecology. One of the bits of work that I started on when I was a postdoc in Australia, and it was a 10-year gestation, but something that we ended up being very proud of, was looking at the early evolution of flowering plants from the point of view of trying to solve the great mystery of why early flowering plants, which are the most diverse plant group on the planet by order of magnitude or two, and also the most recently evolved group on the planet. So there's a bit of a mystery about why they appeared, why they diversified, why they're so diverse compared to seed plants that are not flowering plants, which are conifers and cycads, gymnosperms. Try not to be too technical. One of the mysteries was that when flowering plants first appear in the fossil record 180 million years ago or so, they had very small seeds. The seeds were a couple of orders of magnitude smaller than anything else that was around in the time. The only other thing that had smaller propagules were spore-producing plants like ferns and mosses, which was kind of puzzling. So why couldn't the other sorts of plants produce small seeds, and why should it matter anyway? Cost-benefit theory predicts that the cost of producing something should be proportional to the benefit. And in the case of seeds, the benefit is the bigger the seed, the larger the seedling it can produce, the more competitive that seedling is, the more likely that seedling is to grow and survive. So producing small seeds is risky unless those seeds can be produced very cheaply. So we set out to test whether you could actually detect any difference between flowering plants, and I'm sorry this is messy, but I will talk through it. I'm trying to keep away from statistics. Sorry, Richard. I'll talk through it. So we're testing with existing, like modern day flowering plants and conifers and cycads, whether you could detect any difference in how costly it is to make seeds. So if we look at this first graph here, I feel like I'm starting to give a second year lecture now. Yay! Now, so the direct cost of reproduction is the investment in the seed itself. Okay, the accessory costs, the indirect, are all the other things that have to go into making that seed. So producing stalks, flowers or cones, surfaces that capture pollen, protecting it from herbivores and all those dispersal structures that go along with seeds, and also the cost of failed pollinations. That has to be taken into account. If a species or a group of plants are able to produce seeds of different sizes but where cost is proportional to benefit, i.e. seed size, then the slope between these would be 1. So it would be no more or less expensive to produce big seeds or small seeds because the costs were proportional. If the Now I'm realising I should have bought my reading glasses. If small seeds are more expensive, the slope would be less than 1. If small seeds were cheaper to make, so the slope would be more than 1. So we're talking about whether there's economies of scale in that bigger seeds are cheaper, which is the lower slope, or whether changes in reproductive allocation could make small seeds cheaper. So that's the theory. This is the actual data, and this is just oh my God, it's disappeared. What happened? Oh good, it disappeared here. Lovely IT guy just put his thumb up. We're good. Why is it disappearing? Yeah, it's here. Thank you. Don't do that to me. So I'll stop touching anything. So the other side, those two graphs, those are just small snapshots of the data that we found that we were able to show. So the red lines are the gymnosperms, conifers and cycads, and that slope is significantly flatter than 1, so small seeds are more expensive. The blue line are the angiosperms. This is the top graph. Am I allowed to touch it again? Hopefully it won't disappear. The blue line is the flowering plants where costs were proportional to benefit, and so there was no disadvantage to producing small seeds. So that was great, because then we could see that there was something going on with the plants that preceded the flowering plants, and that they weren't able to produce small seeds as efficiently. And then the bottom graph, where the slopes are actually the opposite, tells the whole story. So the red line, the gymnosperms, there was high rates of abortion and wastage of resources for small-seeded gymnosperms. Because they don't have self-incompatibility, there's a whole pile of other things that mean that reproduction in gymnosperms is quite inefficient. So it's not viable strategy for them to produce small seeds. And so then when flowering plants came on the scene and were able to, through just a simple change in the way resources were allocated, could lower the cost of seed production, they took over the understory and invented the weedy lifestyle. And this basically explains why this particular group of plants took off and diversified. So we're very proud of that piece of work. Coming back to New Zealand, I had been exposed to the Australian flora, and that was a shock because it is so different. Australians are obsessed with ants and fire, but also the flowers are very different, the animals are very different, and so I wanted to understand what was going on with New Zealand plants. I'd already been part of a body of work that showed very clearly that not only is seed size part of the important to the early evolution of flowering plants, but seed size also constrains so much more about a plant species ecology, what it does in the environment. So seed size in itself limits how seeds can be dispersed. Large seeds are not going to be able to float on the air very well, so we see very clearly that once a species is it's locked into a large seeded strategy, it then needs to rely on dispersal by animals because there are a few other ways of getting that seed around. But the reverse also works as well. So comparing New Zealand to Australia was very informative because in New Zealand most of our passerine birds have relatively small gapes. The kiruru is one of the few birds that can eat big seeds, and our fleshy fruited plants actually have very small round berries, and if they have big berries they're cold capsule shaped, and the width of those bigger berries is related to the gape size of our largest birds. You might ask, what about moa? Well, I'll show you. This is a fruit, an Australian Plankinella that's dispersed by cassowary. It's about the size of a gobstopper. These are New Zealand Plankinella. There's no evidence in the most recent subfossil remains of plants that went extinct when the moa went extinct. There's a hint of some big seeded, big fruited things in fossil deposits from several million years ago, but that still needs more work. That's something that was found by a student co-supervised between myself and Daphne Lee. But the other cool thing is that New Zealand animals are weird, we know that, and we have all of these lizards which actually here we were able to show in New Zealand are very likely drivers of the evolution of different types of fruits and seeds, particularly pale colours, and that was work that I started on very early when I came here, and it still continues. It's one of the master's students that I co-supervise. So, flowers. Obviously, seeds and fruit, they're products of evolution between plants and animals, but one of my key passions when it comes to trying to understand how that interaction between plants and animals has driven plant evolution is it comes down to floral biology and flowers. And isn't that a beautiful photo? That's one of Carolisa's. Most flowers in New Zealand are actually quite small and white. They're not pretty like that Dolicha glottis. The number of times I've had visitors come here, like botanical colleagues come here, and I take them to an alpine meadow that's in full flower, and they look around going, what? Where are the flowers? I never really understood this until I went to the Rocky Mountains Biological Laboratory in Gotham, and it looks like someone's garden. It's full of delphiniums and things. So New Zealand, we are famous worldwide for having the highest proportion of white flowered species globally, particularly the alpine flora. When I first got interested in why New Zealand flowers were the way they were compared to Australia or elsewhere, I was told that that's because either they're all self-pollinated, there's nothing to see here, go away, or it's because they were all pollinated by flies and flies can't see colour, so nothing to see here. I think it's something about the South Island personality, but if someone tells me that you can't do something, or there's an underdog, then I say, well, blow you, I'm just going to go and find out for myself. So this actually this led to research for many, many years trying to figure out why we have so many small white flowers. Now, in terms of our unusual pollinating fauna, the key difference, and I'm looking for Jenny in the audience, the key difference is we don't have any social bees, and Jenny loves social bees. We have lots of solitary bees, who are very active as flower visitors. We have lots of flies, particularly hoverflies. We have Californidae and Tachynidae, and there's a whole bunch of other things that visit. The interesting thing, though, is that you can't blame the lack of social bees as the reason why we don't have colour, because solitary bees have colour vision just as well as social bees. The reason why there's this assumption that the lack of social bees might lead to lack of flower colour is derived from patterns that can be seen in the Northern Hemisphere. So Lorna Little here, PhD student of mine some years ago, actually looked at the Pan-Arctic flora. In this lovely graph here, the purple dots, the size of the dots indicate how common purple and blue flowers are in those particular areas. And in the second one down here, the size of the black dots indicate the species richness of bombus, bumblebee species. And there's a very nice relationship between the more species richness there is in the bumblebees in the Pan-Arctic area, the more purple-blue flowers you get. So if that works there, does that explain why we don't have purple-blue flowers or coloured flowers here in New Zealand? Well, no. Through a number of different master students and PhD students, who are all wonderful, intrepid women, willing to sit on mountaintops staring at flowers for long, long periods of time, we were able to establish that our pollinators, our native solitary bees and our hoverflies were very good at actually telling the difference between different flowers. If you painted the flower the wrong colour, they knew. They would show a different preference and they would change their behaviour. So this is the picture here. This is Marcia Bischoff's PhD, where she changed the colour of flowers to look at behaviour of these pollinators. We also were able to gain spectral reflectance data of many flowers and look at how flowers appeared to a bee, because obviously bees have a different colour vision to us. They're trichromatic, but they have receptors in the UV, and they lack receptors at the long wavelength end of the spectrum. And if you plot a community of flowers that look very similar to us within bee colour space, the dots are far enough out that theoretically the bees should be able to tell them apart. So lack of colour cannot be explained by lack of colour sensitivity in the pollinators or lack of social bees. Vicky McGimpsey's masters on Euphrasia actually offered a key insight, in that Euphrasia worldwide is known to vary in colour, and Vicky very carefully was able to show that they actually express anthocyanins when they're pollinated, which is counterintuitive, and then the pollinators know to visit white flowers. So that very limited expression of anthocyanins, actually very conservative resource resource conservative strategy in an environment that's unpredictable, where pollinator visits are low. Saving the pigments, not using pigments, or only using pigments as a signal when pollination has occurred is actually a very good strategy for our mountain plants. And that's where we ended up in saying that lack of pigment is because of the environment and the unpredictability of pollination, not because of the pollinators themselves. But paradoxically, there are amazing coloured flowers down in the subantarctic islands, which is totally weird because there's not even any bees there. There are flies and there are moths and there's a bunch of other crazy things. So I was very lucky, very lucky to go down there 4 or 5 times now. And our work attracted attention from the BBC, who also took us down and paid for things, which is great, so they could film some of our work. Colour in these flowers is serving a different purpose, and infrared photography revealed that these dark and deeply coloured flowers, it's not the pigment or the colour itself that's having anything to do with that plant-animal interaction, it's the heat. The infrared, the concentration of long-wave radiation with a dark surface causes the flowers to heat up, which increases the speed at which pollen is released, but it also creates this lovely warm basking space where flies are attracted, and that seems to serve pollination effect. So that was world first, and also tied in very nicely with some of the work that's been done on giant alpine lobelias and things in Mount Kilimanjaro and stuff like that. We also, when we were down there, we were able to uncover a whole raft of other novel interactions that no one had ever looked for before, because so much of the activity on the subantarctic islands has to do with feathered things or furry things, which are not so important. We documented for only the second time worldwide that an orthopteran, a weta, could pollinate. There's the smoking gun. We found, so this is anisotomy, which has separate male and female flowers. We found weta with pollen on them on female flowers, which means they must have been on a male flower, climbed all the way down, because they don't fly, climbed all the way down, all the way across, all the way up to a female flower, and they still had pollen on them, so capable of transferring pollen. This guy is one of my favourites. This is a flightless fly, a wingless fly. We should call a walk, maybe. But look at its legs. It had these big forward-facing eyes and these enormous legs, and it jumped from flower to flower and was carrying pollen on its body. There's a shellless snail. It's a crazy place, a totally crazy place. Shellless snail that was, it had pollen on the outside and the inside of it. And Max Buxton, who is hopefully watching, sorry, I'm getting a little, I haven't had coffee, honestly. Max Buxton, who is a moth expert, and he's now just finishing his PhD up in Hamilton. He was able to document that moths were incredibly important in these environments as well. So that was super exciting. But working in these cold climates, you come very quickly to realise how fragile any ecosystem is that depends on snow, depends on cool water or cool temperatures. And all this time in working in these cold climates, I was very aware of how the changes associated with the rapidly warming planet were affecting these ecosystems that I had spent so much time looking at. And 2016 was a bit of a pivotal moment. That's when the first global analysis was completed that showed that the hottest year on record with the planet nearing 1.5 degrees warming, and we've since overtaken that landmark. It's also around the time where I became involved with OCNET, the Otago Climate Change Network, which then morphed into He Kaupapa Hanganga, Otago's Climate Change Research Network. So that also changed the direction of some of my research. I was part of a team that was led by Professor Cass Dickinson, who said she'd be online. Hello, there you are. And where we actually tried to test what would happen if there was less snow in the environment. What would happen to these alpine plants? So this is the Rock and Pillar Range. This will bring back memories, Kath. Where the different environments in these alpine ecosystems experience very different levels of snow, and the patterning of the snow lie over winter is actually a very strong driver of vegetation pattern. The deep snowbanks especially are completely covered in snow, and snow matters in winter because it's actually warmer under snow than it is being exposed where there's frost and wind, which is very damaging. An extensive reciprocal transplant experiment enabled us to examine the effect of taking whole turfs of snowbank communities and putting them out in a simulated future warmer climate. And the end result was that it was very clear, if you look at these pie charts here, this is the late snowbank species when they've been transplanted to areas with less snow. The black chunk and the red chunk are species that disappeared, went extinct, and the red chunk are species that were on the way out. So losing that snow cover was going to be profoundly, having a profound effect, particularly on those snowbank species. Looking at climate change impact, documenting climate change impact is something that researchers have done for many decades, and so much information has gone into the IPCC reports, and yet so much of that evidence hasn't had much of an impact. What has become increasingly important is understanding how we can use the knowledge we have to mitigate climate change and to adapt to climate change. And so worldwide there was at the time, and there still is, a growing interest in carbon sequestration. So at about that same time I became involved in the 1 Billion Trees program, and looking at how we can reforest to sequester carbon to help New Zealand meet its international obligations to reduce net emissions. The 1 Billion Trees program was launched in 2019 or 2018, and it's a 10-year program, so by 2028 the aim is that 1 billion trees have been planted. This is a current update, so we're kind of on track. And but the balance of native versus exotic trees is perhaps not where a lot of us would want it to be. So the 1 Billion Trees research program that Richard referred to has dominated my life and my family's lives for the last 5.5 years, 6 years. It has been an incredibly rewarding, incredibly frustrating piece of research, but it has really opened my eyes to the possibilities of engaging with private sector, with public sector, with landholders, and we were able to do experiments at a scale that just wouldn't have been possible. We had access to tractors and diggers and all manner of agricultural machinery, and the help of wonderful people like Russell Hamilton and Huntley McGregor, and also the guilds at High Peak, enabled us to explore questions around how to accelerate native forestation that we wouldn't have otherwise been able to explore. That box there is a whole list of students and collaborators that have been involved. I have kept on adding names as I've remembered people. I apologise if I've left anyone out. But the key take-home messages that have come out of that is that in order for native reforestation to become equally efficient compared to exotic reforestation, we need whole system thinking. At the moment it is still more expensive to plant natives or sow natives, so thinking about systems in their entirety is important if we want to create landscapes that will promote native biodiversity going on into the future. Some of the key take-home messages and some of the most exciting work that's come out of this has had to do with the below ground ecosystem, because in degraded ecosystems where it's pasture or retired pasture, it's an alien microbiome, and introducing native species into that is not going to be helpful unless you rehabilitate the soil microbiome as well. Melissa Strawson was a master's student on the 1 Billion Trees program, and her work demonstrated that Manuka seedlings inoculated with natural Manuka duff, which is the organic layer that contains the mycelium, were much more drought tolerant than if they weren't inoculated with duff. Dr. Laura Van Galen, who is an exceptional researcher who created so much new knowledge around the relationship between beech forest and ectomycorrhizal fungi, also experimentally was able to show that colonisation on the roots of beech seedlings was related to how well they grew and survived. And work where we actually use molecular techniques to look at the diversity of mycorrhizal fungi on plants that we'd planted or sown really shows the level of the problem. So in this graph here, the two different coloured bars are just two different types of samples that were analysed for amplified sequence variance, which is just kind of equivalent to taxonomic diversity of ectomycorrhizal fungi, or not taxonomic. David will probably slap me if I say that. But here, these two sets that are really quite high in their diversity, this is natural Manuka shrubland. These lower sets here, these are plants that came from Marissa's experiment that we then planted out into the field. I had expected, this was several years later, I had expected by that time they would have accumulated more diversity, but they hadn't. So clearly, and they hadn't grown very well either, so clearly there was still some gaps in our understanding of how to facilitate these native plants in this foreign environment to get the mycorrhizae they need so we can quickly grow native forests. And you can see the pine forest down here. This was a pine forest, and this was an area that had been in pine forest, very depauperate from the point of view of the mycorrhizae that native plants need. So new native reforestation requiring systems thinking, and this decision diagram I produced for MPI because it became very clear that this was the kind of information that was needed to feed through to those communities that were wanting to do native reforestation. We had all of this very academic data on amplified sequence variance and all this great stuff and statistics, but to communicate it to the people that needed to know, had to change my brain outlook to put it in a way that really delivered the practical outcomes that people needed. So MPO was really quite pleased when we got to the point of a decision tree, because that's what they needed to communicate to their stakeholders. Right, which brings me to, I guess, the area of my research that has been the most challenging, but also I feel the most impactful, and that's where, thanks to associations with Professor Zara Walton, sitting there in the audience, and Dr. Victoria Kauhui in economics, stepping way outside of my comfort zone into economics and social science and thinking about policy and people. And I found being able to bring my botanical kete to the table, along with economics and business thinking and social science thinking, enabled us to produce something that was truly innovative, where we could bring insights to how policies might produce perverse incentives that end up with bad environmental outcomes, even though the policies themselves might be well-meaning, the outcomes might actually be environmentally degrading. An example would be freshwater policy which restricts grazing, but a different policy would allow pine planting, and that could actually be a worse environmental outcome. And afforestation, we know afforestation is very rapidly changing farmland from food production into tree production because of the policy drivers of the economics. It's going to create very different fire regimes, and we're not ready for that. So botanical knowledge about how we can manage fire in a landscape, what kind of trees should go where, it becomes really, really important in informing policy. The other aspect was diving into understanding how farmers are feeling about the policy landscape when they feel like they're doing the right thing, and yet they're not getting any credit for that. In fact, it's coming as an opportunity cost because they're actually taking away some of their productive land in order to do the right thing and restore a wetland or plant trees. There are opportunities out there. There are models worldwide where people can be rewarded for biodiversity or ecosystem services, but implementing those currently in New Zealand is challenging. Hopefully it will come at some stage. So I want to kind of wrap up, and I'm going to use this whakatauki, this waiata, to illustrate how I have grown, I guess, and how I feel like I still really don't know that much. I was first taught this waiata when I was part of a team that was investigating traditional plant use by Māori, particularly weavers, and we were going on a hikoi, visiting different marae, meeting with weavers. I was super naive. I had no idea. I really had to be taught. I was very lucky that the late Rua McCallum was a mentor, and she basically slapped me when I got it wrong. But when I first learnt this waiata, hutia te rito o te harakiki kehia te komako I ko. If you cut out the heart of the harakiki, where will the bellbird sing? And I was like, oh, sweet, it's about plant and animal interactions. It's about pollination. Because the rito is the central young leaf. It comes from the apical meristem. When the fan of the harakiki switches to being reproductive, that apical meristem becomes floral. It produces the kaladi that makes the flowers. And then the whole fan dies. If you take out that apical meristem, you lose that ability to produce a flower. What I didn't realise, but what I learnt, is that anything in Te Ao Māori has layers. And plant symbolism is so much at the heart of culture, Māori culture, but any culture around the world. And the harakiki, plant harakiki fans are very much symbolic of family and society. So taking out the child, taking out the young people, losing engagement with young people kills off the heart, the soul, the song of the family. So that's why the second half of the waiata is about people. I could never understand that initially until I understood that engaging plants and people together is what is good for the health of society. So I've been very fortunate to be mentored by, as I said, the late Rua McCallum, and collaborated with Sila Wehi, Marion Johnson and Dr. Kerry Hartley, who have pushed me outside of my comfort zone and taught me to look more at how people engage with plants, because that's how you translate your science, your data, which we're all very comfortable with, into actual impact when you get people to connect and care. So, finally, a plug for things that are happening in the future, and this is again where the botanical knowledge, taking my botanical kete to a table, can produce exciting results. These are two movements that are coming out of the private sector that I reckon you guys would be interested in looking up. A wonderful wahine, Chanel Gardner, is leading a charge to apply for UNESCO Biosphere status for the Dunedin sort of inland Otago region. If you don't know what a biosphere reserve is, then look it up. And Catherine van der Meulen is leading the growth of a vision to, basically create a vision of what Aotearoa New Zealand could look like in 100 years' time if we get it right, if we engage with nature-based solutions, if we act in a climate positive way. And this is modelled on a program that the Netherlands launched 5 years ago, which has generated significant new collaborative research. So these are exciting things, and I'm really excited about this because I can take my botany and bring it to the table with all of these other innovative people and create something wonderfully multidisciplinary that will have an impact in the future. So, lots of pictures of people. I was rummaging through my files trying to find pictures of people, and there were people that I didn't have pictures of. Some of those people are no longer with us, that's the top row, and I really do honour the memory of those people who have significantly influenced me in my life. In the middle, there's an adventurous club of amazing, intrepid women and other people who've come out and done crazy field work in the sun and the rain and the snow. And of course, my extended whānau, the Botany Department, so grateful for all your support. And it is a privilege to be part of this department in this university. So, I shouldn't forget my wonderful sons, Tor, who are trained for field work from an early age, if anyone wants to employ them, and my long-suffering partner, Ash, who has fed the pooches and the ponies while I have been in the field, and you have supported me to realise my dreams. And I'm going to cry now. So I will stop and say, ngā mihi nui. Thank you.
Thank you, Janice. Ko David Oliver Tahoe. I'm the head of the Department of Botany, so I've got the privilege of wrapping this up and there's no questions. Yeah, that was an absolutely wonderful talk. When you got introduced by Professors Rua and Barker, they talked about all your achievements and everything, but what they couldn't have expressed was how engaging a speaker you are. And as soon as you started talking, we could see why we're so lucky to have you in the department. That enthusiasm is passed on to the next generation, and that's why we're so lucky, really. So you also showed really nicely how the study of botany is just like the seed that can go into a lot of different places. And your talk started from the really small to the global, and that's what your research has been. And got to say, I'm jealous of that part because I'm a small picture person. I'm not very good at being a big picture person, and you've managed to transcend that, so that's really good for us. So congratulations from me and from the department on your promotion, and thank you for an absolutely engaging, wonderful talk, and all the best for the next future. So the final thing really is to say thank you from all of us and to invite everybody to the staff club for refreshments. And what's that? Yeah, I didn't actually get it, so they didn't give it to me, so I'm gonna, there is a thing. Oh yeah, we'll do that, yeah, all right, yeah, do you want to do that now then? Yeah, all right, yep. I'm gonna have a wire, so, yep.
Guys, come on, stand up.
Okay.
Who's gonna start? You start.
Is that Hutia Ture Huti. You say it, you do it. Yeah, you do it.
Hutia te rito, Hutia te rito hara ke hia te komako e ko, ki mai ki ahau. Hei aha te mea nui, hei aha te mea nui o te ao, maku I ka ki atu. He tangata, he tangata, he tangata, he.
That was